Hi everyone, I'll now be going through the TE for renal system. Okay, very broadly again, I would like to recap some of the key things you want to look out for the renal system. In the renal system, we tend to ask you questions about the structure of the renal system. What do I mean by that? That means we are looking at the structure of the broad structure of the kidney, right? All the different parts, the blood vessels that go in and out, and the microstructures like the nephrons. That means you need to know all the different parts. Okay, and from these questions, we typically ask you to identify the parts, or we may ask you to trace the pathway of blood or filtrate. Other than that, other than structure, we may ask you about urine formation. So what do I mean by urine formation? We will be asking you about various processes involved. Number one, ultrafiltration. Where does this take place? What are the adaptations of the nephron that allows for this? May I also ask you about selective reabsorption? So under selective reabsorption, what are the adaptations that allow for various parts of the nephron to reabsorb substances? Okay, last but not least is secretion, but this one we rarely ask you one. And third, we often ask you about homeostasis. What are the homeostatic functions of the kidney? And more specifically, we look at osmo regulation. So, for example, how does ADH secretion affect the permeability of the walls of the nephron? We may also ask you about how dialysis occurs. Okay, so broadly, these are the three categories of questions. Let's begin first with question 1. If you look at question 1, it's actually a question on structure and tracing the pathway that filtrate takes. So what's the correct pathway that the filtrate takes? It should start off with the proximal convoluted. It should start off with the renal corpuscle. So anything with 6 should be the answer. So these two options possible at the moment. So this is okay, this is okay. After that, the proximal convoluted tubule. After that, you should go down the descending loop of Henle. So now we know D is likely the answer already. Then the ascending loop. Then after that, the distal convoluted tubule, then the collecting duct. So the answer is D. Where can you find the highest concentration of urea? Okay, the nephron? No, that's not the higher concentration. Renal artery. Okay, renal artery, yes, it will contain urea, but it won't be the highest concentration yet. Okay, the renal vein, you should expect on the lowest concentration of urea because it has just been cleaned. The blood has just been cleaned by the kidneys. It's actually the ureter, because the ureter is where all the millions of nephrons funnel via the collecting duct into the renal pelvis and into the ureter. This should be where the concentration of urea is the highest, not the blood. Yeah? So answer is D. Let's look at 3. Which part of the nephron was this transverse cross-section taken from? So this is a cross-section of a part of the nephron, and actually this part over here is all the microvilli. In other words, this is likely the proximal convoluted tubule. So the answer is D. The loop of Henle collecting duct doesn't have this unique microvilli. The Bowman's capsule, also you will not find these structures. Another clue is that, although not very clear, there's actually a lot of mitochondria present. This is required to supply energy in the form of ATP, so that active transport can be used to reabsorb the nutrients. Next, let's go to question 4. Diagram shows a kidney nephron tubule and its associated blood vessels. Where are the blood vessels? The blood vessel is this one over here, or this bundle, and then goes round the nephron. Okay. Which graph shows the correct concentration of glucose present in each part of the kidney? Concentration of glucose. Okay, so the first thing you want to take note is that the concentration of glucose at Y and Z actually should be close to 0. There should be nothing left, because the kidney will, as much as possible, reabsorb all of it at X, at the proximal convoluted tubule. Okay, so you want to search for a graph where Y and Z is low. Okay, here B, Y and Z is very low, like 0. All the reabsorption should take place at X, so you should see a dip down X, right? B is the best answer at the moment already. W is where you'll find the highest concentration of glucose, because that's where blood is first filtered, and even the glucose will filter into the Bowman's capsule. So the best answer is B. It cannot be C, because it implies that even at Y, the loop of Henle, there's still glucose left, and at Z also still have glucose left. Perhaps this is possible for someone whose kidneys are damaged. Nephron's not quite doing its job anymore to reabsorb substances fast enough. This might belong to a diabetic patient, C, right? But so a healthy kidney should be B. Let's look at question 5. Which of the following statements is true? Okay, that means 3 of the answers are false. Cells of the PCT possess a brush border of cilia to aid in selective reabsorption. Why is this wrong? Actually, it's villi, not cilia, yeah, or microvilli. Next, increase in level of ADH will lead to increased urine production. That's not true. If there's more ADH, that will also mean there will be more aquaporin. More aquaporin means more water reabsorbed. Then there should be less urine formed. C, the distal convoluted tubules regulate blood pH by controlling H+ secretion and HCO3 reabsorption. This is true. D, the ascending loop of Henle is permeable to water. This is not true. It's permeable to solutes, like the salts, but not to water. It is the descending loop of Henle that is permeable mostly to water. And that's one of his adaptations. Therefore, our best answer is C. Let's look at question 6. Which statement is true when a person is dehydrated? Person is dehydrated, that means there's not enough water in the system. If there's not enough water in the system, the blood osmolarity would be high. Okay, so if the blood osmolarity is high, more ADH is secreted by the hypothalamus. Okay, this statement is not correct because it's produced by the pituitary gland, not the hypothalamus. The hypothalamus role is to detect the change in blood osmolarity. So there are receptors in here, but it's not the structure that produces ADH. D, more aquaporins are inserted into the walls of the loop of Henle. While this part is true, not the loop of Henle. It should be the collecting duct, as well as the distal convoluted tubule. More water is reabsorbed at the distal collecting duct. Okay, distal means the collecting duct. That means the later parts of the collecting duct. This is possible. More water is reabsorbed through active transport. This is not true. Water is reabsorbed through osmosis. If we want more osmosis to occur, then we need more aquaporins. So C is the best answer. Last MCQ question. Which statement correctly describes hemodialysis? So let's look through the options. 1. Blood is drawn from a vein and returned to an artery in the patient's arm. This is not true. Actually, blood is drawn from the vein and also returned to the vein. Blood is drawn from the artery and then after returned to the vein. Okay, so it's not drawn from a vein and returned to the vein or to an artery. Statement 2. Composition of dialysis fluid used consists of glucose, amino acids, ions, and urea. Okay, dialysis fluid shouldn't have urea. You don't want it to have urea so that you can establish a concentration gradient. The blood that contains urea that that will end up having the urea diffuse out into the dialysis fluid. 3. Direction of blood flow is similar to the flow of dialysis fluid to maintain concentration gradient. That's not true. Blood flow and dialysis fluid should flow in the opposite direction. Last but not least, waste products such as creatinine will diffuse out of the tubing into the dialysis fluid. That's true. That's the whole point of hemodialysis. We want waste products to diffuse out of the tubing into the dialysis fluid. So the best answer is B. Only statement 4 is correct. Let's go to the structured questions. Structured questions is a classic example of testing you the structure of various parts of the kidney, in this case the nephron. They show you an electron microscope image showing the renal corpuscle of a mammalian nephron. They already told you that this particular cell over here is the podocyte, and this is the inside of the capillary, and that this is the Bowman's capsule. So with all this information, you must be able to make sense of what's what. So if this is the podocyte, this is the capillary. In other words, likely what you are viewing over here, okay, let me highlight it in yellow. This region over here, the bottom part over here, is likely the walls of the capillary. Okay, the endothelial wall of the glomerular capillary. If you look very carefully, you can in fact see the fenestrations. Here is a fenestration, here's another fenestration. Lots of gaps, right? And that makes sense. After the glomerular, the fenestrated glomerular walls, the next layer you should see, you see this thin layer over here, this is likely the basement membrane or basal lamina. After that, since this is the podocyte, right, that means these are the pedicels, and they interlock to form all the filtration slits. So this is an example of filtration slit. These are all filtration slits. Okay, so name the structures A and B. A is pointing to the basement membrane or basal lamina. What is B? What are all these things pointing to? These are all the pedicels, and in between the pedicels are all the filtration slits. They ask you to draw an arrow to show the passage of fluid out of the capillary, and then the fluids will have to flow out of the capillary, right, through the fenestrations, which are the holes in the capillary, and then after that between the filtration slits of the pedicels. So that's an example of an arrow you can draw. Explain what is required for ultrafiltration to occur. Asking you for the adaptations that allow for ultrafiltration to occur. So first one you can mention is that you need sufficient hydrostatic pressure in the glomerulus, right? And how is this achieved? This is achieved by having an afferent arteriole with a larger lumen diameter compared to the efferent arteriole. Efferent is the one exiting after the glomerulus. Next, you should have a presence of a glomerular filtration barrier, and if we want to elaborate on it, it means that first one would be that the capillary wall or the endothelium of the glomerulus is fenestrated. You want to mention the basement membrane. You want to mention the filtration slits formed by the pedicels of the podocytes. Last but not least, you want to mention the slit membranes. All of these provide a size selective filter that only allows small molecules to pass through. Okay, and that is what is required of ultrafiltration. It's a very typical question. Next question. Okay, they show you table 9.1. Ask you to write the missing concentrations in terms of X and Y of protein and glucose in the table above. Okay, so they show you three different substances: large protein, glucose, urea. In the glomerulus, glomerulus means still inside the blood capillary. You will expect to find, they already told you, concentration of X, Y, and Z of these three things present inside the capillary. So after ultrafiltration has occurred from the glomerulus into the blood capillary, into the Bowman's capsule, what can you expect to find inside the Bowman's capsule? You will find that there will be 0 of large protein because that stays behind in the glomerulus. You'll find that glucose and urea will both pass through, so the concentration is the same. By the time it hits the distal end of the proximal convoluted tubule, that means distal means far away. That means it's nearing the end of the PCT. What will you find left? You should still find no large proteins, and this time around you'll find less glucose present, or 0 this time. They should be all reabsorbed by active transport. You find instead that urea concentration increases as we go along the tube, the nephron. That makes sense because more and more water is reabsorbed, so the concentration increases. So name 2 substances not listed in table 9.1 that will be present in the glomerular filtrate. So you could mention mineral salts, amino acids, vitamins. These are all small enough to pass from the glomerulus into the Bowman's capsule. Next, briefly explain the difference in the changes of the concentration along the nephron for the protein and glucose. So how do you explain why glucose starts off with X amount in the glomerulus, but then you don't find any in the Bowman's capsule? How do you explain that? Well, because the proteins are too large to pass through the fenestrations of the glomerulus. It's also too large to pass through things like the filtration slits or the slit membrane. So these are things you can mention. What about glucose? You can find glucose in the Bowman's capsule, but you don't find it anymore later on in the nephron. 2 things you can mention. One is that glucose is too small, and therefore it can pass through, and also it will be reabsorbed. So glucose is small enough to pass through the filtration barriers, but good thing is actively reabsorbed back into the bloodstream. That's why you find 0 amount of glucose later on. Next, let's look at D. Explain how the cells that make up the PCT may be specialized for their function. Okay, so this is a question of adaptation. So you can mention that the epithelial cells of the PCT has numerous microvilli. This increases surface area for selective reabsorption to occur. That's one adaptation. Another adaptation you can mention is that they have a lot of mitochondria, provide energy for active transport. You can also mention the presence of many membrane transport proteins that allow for the reabsorption of specific substances. Any 2 of the 3 will give you the mark. Next, E. Suggest how having a low blood pressure may affect the volume of filtrate in the Bowman's capsule. Okay, so they ask you what will affect the volume of filtrate. If you have low blood pressure, this will reduce the hydrostatic pressure at the glomerulus. That means there will be reduced outflow of fluid from the blood capillary into the Bowman's capsule. This will result in a reduced volume of filtrate in the Bowman's capsule. Concentration does not change, right? You can change the volume of filtrate, meaning how much fluids will pass through the filtration barrier, but the concentration remains the same. Last question. Alcohol inhibits pituitary secretion of ADH. And overconsumption of alcohol leads to a hangover, which is characterized by thirst and frequent urination. So explain how alcohol may bring about the symptoms of a hangover, the symptoms being thirst and frequent urination. So in a sense, what's happening here is that alcohol kind of artificially makes your body go into a state when you are overhydrated. So why I say so? Because it inhibits the secretion of ADH. Right? ADH is produced when you're dehydrated. Osmolarity is very high. If you have a lot of water in your system and you're overhydrated, there'll be less ADH produced. So that's kind of what alcohol is causing. So ADH makes cells of the collecting duct permeable to water. Okay, you want to mention what's the function of ADH. You can elaborate by saying that it makes it permeable to water because of the aquaporins. So if you inhibit ADH secretion, that means the collecting duct or the distal convoluted tubule will become less permeable to water because there are less aquaporins. This will also mean there will be less reabsorption of water and result in a large volume of urine form. This explains the frequent urination. Because the blood osmolarity increases, this will stimulate the osmo receptors of the hypothalamus, and this could stimulate the thirst center of the hypothalamus, causing the person to feel thirsty. Okay, so one may be asking, how does larger volume of urine form lead to increased blood osmolarity? If you end up urinating more than you actually need to, it will cause your blood to have less water, right? And if there's less water in your blood, then the blood osmolarity will increase because you're artificially being forced to urine a lot more, simply because your body is not reabsorbing the water. Okay, and then that will lead to the thirst. Okay, so that is the answers for this TE. If you have any questions, please feel free to let me know. You can find out what each question is trying to test you by looking at the success criteria related to each question. Then you'll know whether you are able to understand each success criteria or not. Use it as a gauge for your understanding. Okay, thank you.